So very recently, neuroscientists, computer scientists, and scientists from around the world have created one of the most comprehensive wiring diagrams of the brain. In other words, the study of how neurons connect to each other is called connectomics. It's called the field of connectomics. And this comprehensive wiring diagram is called the connectome or the brain map. And we will be using this very fascinating data set to uncover new insights about the brain. And together, Arushi and I, and on behalf of our faculty partner, Professor Eyal Gruntman and Anreiter, want you all to be a part of our learning journey to use powerful connectomic tools to visualize and analyze the connectomics data set. So in other words, we will actually be using two powerful tools in analyzing, a large-scale brain data set. The first one is called Neuroglancer. You use Neuroglancer to actually visualize this complex brain data. And think of the brain as like a 3D image, right? But within this 3D image, you have different slices of the brain. And within this brain, you have like 50,000 neurons and so on. So actually, a group of scientists have created a very cool platform to actually visualize neurons and look at its structures. And today, Arushi and I will be showing you how this Neuroglancer is such a cool tool. Next, we'll be also using neuPrint. So neuPrint is a tool for querying neuronal connectivity. But you might be like, why do we need to, like, how is it fascinating to actually query neuronal connectivity? Well, just to keep in mind, the optic lobe, which is the area related to visual processing in the fly, this optic lobe alone has over 53,000 neurons, and they have classified it into 700 plus neuron types. And together they form 49 million synapses. In other words, there are 49 million connections. So as you can see, it's such a massive data set, and covering a small part of this neuronal connectivity is something that connectomics researchers, neuroscientists do every day. It's such a powerful tool to like gain new insights about the brain. And by studying neuronal connectivity alongside with actually visualizing how the neuron is in a 3D space, you can learn so much about the brain. An analogy I would like to use is like, you diving into a vast ocean to find how thousands of species like, plants and fish interact with each other. And, I would be doing the presentation just to give you like a guide, and Arshi would actually be doing a live demo. But what's the challenge for the competition? And I will come back to this again and again, but just to give you like a goal of what we'll be doing. So the first step is we'll be choosing a neuron type. And choosing a neuron type, we can start with actually using the tool, which is the visualization Neuroglancer tool. But there's actually different ways to pick a neuron that you like. And again, Arushi will sort of show how to do it. The second step is once you pick a neuron type, you will be exploring neuron morphology. In other words, you will be exploring how the structure of the neuron, you'll be exploring where in the space of the brain does this neuron traverse? Like, does it go from region A to region B? And where does it take input from? Where does it output to? Those are very important in informing how this neuron work together to, induce a behavior or process of information. Step 3 is to, once we visualize the neuron type, we will now analyze the connectivity using neuPrint. In other words, we want to see this particular neuron that you choose, how does it connect to other neurons? From which neuron does it take input from? And to which neurons does it output towards? Okay, so we'll be analyzing connectivity using neuPrint. And lastly, you will combine what you explore using the connectomics tools, and then you will actually go search the literature, and together you will try to propose how your particular neuron process certain visual information or process other information, and in turn, how does that contribute to a behavioral response? So I know some of this might not make sense right now, but we are gonna spend the entire presentation talking about this. In the end, once you do all of the research, you will submit two final deliverables. The first is a creative piece. We want to see that you're having fun with this project and that you actually can communicate what you have learned to a public audience. So you can create a creative piece. This could be a video, art, post, infographics, and whatever you like. It can be a poem, it doesn't matter. But the idea is you create a creative piece. The second part is you write a written research report. So it'll be like a 500 to 1,000 words written research report on a specific neuron and how you use like the connectomics tools to gain new insights about how neurons contribute towards inducing a fly behavior or processing certain information. Okay, all of these are in our guide, but I want to point attention to this particular thing, which is the early feedback opportunity. So our final submission deadline is December 1st of 2025. But for everyone who, try and actually do the connectomics research and try to write a report and do the creative piece, if you guys do it if you submit before August 31st, we would actually give you feedback and so that you can improve upon it and submit a better version by the final deadline. And we will announce the winners January 2026.
Hi everyone, I'll be giving a Neuroglancer demonstration of a neuron interest. And to get started, firstly, you have to open Neuroglancer, and how you can do that is you can go to our applications material Google Drive, and if you go to point 1, which is our handbook, and if you click on it, and you scroll down, you can see that we have important links, and you can click on the Neuroglancer one, and it will bring you there. So just making sure we have the optic lobe, Connectome data here, and everything seems right, so we can get started. And the neuron I'll be demoing with today to kind of simulate what this journey might look like for you guys is LPLCD. Something to keep in mind is make sure you have the capitalizations in the right place. It does matter, and if you don't follow those rules, you will not get your neuron. So I'll click on the first one Neuroglancer gives me, and here we are. So the purpose of this exercise is, well, firstly, to document the morphology of this neuron you're interested in. So to start off, let's click on ROI, and this one gives us the neural shells, the optic lobe. Now, if this is a bit confusing and you kind of forgot what each stands for each because there are no labels, you can go refer to our starter guide, and you can scroll down and you'll see that we have actually screenshotted this image and labeled these for you guys. So you can see this is the lamina, this is the medulla, this is the lobula, and the lobula plate. Now that I know, okay, this is my LPLC2 neuron. It is in the lobula plate. But maybe for some neurons, it's a bit hard to tell where their arborizations are going through. So one way to have a look at that is you can click on this pre-syn and this post-syn option. So the pre-syn is the sites where this neuron is sending its outputs, and the post-syn is the site where it's getting inputs from. So like pre-syn, it is presynaptic. This neuron is presynaptic to its outputs. Post-syn, this neuron is postsynaptic to the inputs it's getting from. So you can see most of that is localized within the lobula plate and the lobula. So its arbors are concentrated on those regions. So arbors also meaning the branching pattern of this neuron. Now, since I've honed down, LPLC2 goes from the lobula and the lobula plate, and that's where most of its arbors are localized. You can now dig in a bit deeper and find what layers of those two neurons it goes through. Since we, honed down that, okay, lobula plate is one of them, you can go to the side panel here and click on LOP layers. So if you click on the eye, you'll get LOP layers. And if you click this, you'll get the 4 layers of the lobula plate. So you can click on the eye, and it'll show you all the layers of the lobula plate. Now, in this exercise, you'll have to sort of do a bit of a trial session of where the arbors are localized within these layers. So one by one, I will peel through these layers. So let's remove layer 1. And if I remove layer 1, I see some of its neurites are showing. So that means it did go through that layer. So I'll put it back on. And if we get rid of layer 4, we can see some of its neurites are exposed, which means some of LPLT2's neurites are also going through that layer. So something I can conclude is, okay, so I mean, this one we know is consistently arborized this way, but you could also remove 2 and 3 internally, and you can see some of these arbors are exposed. Anyways, so we know that the lobula plate portion of the LPLC2 neuron transverses all the layers of the lobula plate. Let's see what's going on with the lobula. So I'll just remove this for now, and let's go and look at the lobula layers. So I click on the eye and I open this. So it's 1 to 7 layers, and it loads everything. Might take some time. Do the same exercise. So peel over layer 1, and I see, okay, there are no the PLC2 arbors exposed here. So, well, it's not going to layer 1. Then I'll do the same thing for layer 7. So you see, sort of removing one by one on each side. You can go however you want, but I find this the easiest. Seems like it's not really Again, it's not really exposing any of those neurites, so which is where it has its postsynaptic and presynaptic sites. If you go among layer 6, okay, seems like there are some neurites it's exposing, so it does transverse that layer. Now let's peel back layer 2. Okay, layer 2 seems like it's not really exposing anything, so it doesn't go through layer 2, and if you're layer 3, it also doesn't really seem to be going through layer 3. Then if you're layer 4, okay, some of the neurites are exposed, so it does go through layer 4. So we've deduced here, looking at this, that it goes from layers 4 to 6. So all in all, through this exercise, I have kind of sort of visualized the morphology of LPLC2 and come to some conclusion, which is LPLC2 goes through the lobula and the lobula plate. It transverses all the layers of the lobula plate, however, it only transverses layer 4 to 6 of the lobula. And something else you can also characterize of your neuron of interest is to see how dense are the presynaptic and postsynaptic sites. So again, if I click on this option here and see when I just have presynaptic sites, it has relatively fewer presynaptic sites compared to the level of postsynaptic sites. So there's also something there you can visualize. This is the Neuroglancer demonstration of how to visualize your neuron of interest.
Clematis Research Empowerment Hub. Empowering research for everyone.